Good evening, everyone. Today we'll be discussing the Ebola virus infection cycle, its genome, its structure, and what makes it an inefficient virus, and more to really highlight the importance of this virus. As we know, viruses, pathogens, and other microorganisms are what cause diseases in humans and other hosts, but specifically, we'll be focusing on the Zaire Ebola virus because it was the variant of the virus that caused the major outbreak in Africa not too long ago. We'll also be mentioning the rest of the Ebola virus species, part of the Filoviridae family. As we can see on table 3, this table shows the 5 species of Ebola virus, and clearly the Zaire Ebola virus species has the highest mortality rate compared to the others. In recent years, no other virus has gotten as much attention and brought out as much fear in the public like Ebola virus due to its high mortality rate and worldwide media coverage of this disease. It was even declared a public health emergency by the World Health Organization. This virus infected many countries in West Africa and infected thousands of individuals, with some reported cases even in the US and other Western countries. The bar chart represented below represents the number of cases of Ebola virus disease during the outbreak in the country of Guinea by week of symptom onset during 2014 and Sierra Leone. But in June of 2014, 398 of the 528 total confirmed cases were reported from Guinea. As you currently know, we are living through the coronavirus pandemic. Ebola virus was an impactful virus that gave me, and I bet a lot of you guys, your first insight to how detrimental viruses can be to our health and to our daily lives. The figure summarizes the West Africa Ebola virus outbreak as per September 8th, 2014, geographically.
The Ebola virus is a part of the filovirus family. It's helical and polymorphic. It's enveloped in a non segmented RNA virus. Its genome is 19 kilobases long with 7 genes. The 7 genes are the nucleoproteins, virion proteins 35, VP40, glycoprotein, VP30, VP24, and RNA dependent RNA polymerase trailer. The viral particle is a single strand negative sense RNA molecule enveloped in the lipid membrane. Some target cells of the Ebola virus are the monocytes, macrophages, hepatocytes, and adrenocortical cells. It is considered a zoonotic disease where primates and non primates can become infected and potentially become fatal. Fruit bats are known as a vector host. And transmission to humans can occur through undercooked bushmeats or contact through hunting or prepping an infected carcass.
The following is the infectious cycle of the Ebola virus. Once a healthy person has been exposed to the virus, many through bodily contact, Ebola can target any cell that can facilitate its attachment and entry into the cell. To elaborate, Ebola's glycoproteins must attach to either a C-type lectin, used for adhesion and immune responses, T-cell immunoglobulin and mucin domain 1, TIM1, also known, used for clearance of apoptotic bodies, or tyrosine kinase receptor, used for cell signaling. Once inside the cell, the virus can replicate its 7 viral proteins, which can be denoted here by this diagram, a nucleoprotein denoted by the red arrow, 4 virion proteins denoted by the blue arrow, its glycoproteins denoted by the purple arrow, and its RNA-dependent RNA polymerase denoted by the yellow arrow. All viruses, its infectious cycle can be split into 4 steps: the attachment and entry step, the genome release into the target site step, viral genome and protein replication, and viral exiting. And it's no different for the Ebola virus, as we will see here in the following diagram. So for the Ebola virus, attachment occurs through glycoprotein and some target cell receptor, in this case, a C-type lectin. The virus then enters through the micropenocytosis pathway, also known as the endolysosomal pathway. And upon entry, PI3 kinase is activated, which activates ACT, which activates RAC. RAC inhibits lysosomal binding to the endosome carrying the virion, preventing its degradation and allowing its negative sense single-stranded RNA to be released into the cytoplasm. RNA-dependent RNA polymerase and BP35 form a complex and allows for this negative sense RNA to be transcribed into mature viral mRNAs, or positive single-stranded RNAs. These mature viral mRNAs can then be translated into the viral proteins like VP40, glycoproteins, nucleoproteins, the polymerases, but most importantly, VP24, which can be seen here. VP24 inhibits KPNA1 dependent nuclear transporters, which are crucial for interferon data release into the cytoplasm, which makes the interferons and to induce antiviral states to nearby cells. So with VP24 inhibits this transport and interferons cannot be released. RNA-dependent RNA polymerase and VP35 once again form a complex, and the viral RNA is replicated. This viral RNA is taken to the cytoplasm through actin transport, and then all the proteins along with the nucleic acids are virally butted off into the environment, where this new enveloped Ebola virion can infect the same target cell or go to a new type of cell.
So how do we measure Ebola presence? There are various methods of Ebola diagnostics in the laboratory that are available. Some examples include polymerase chain reaction, ELISA, which is enzyme-linked immunoabsorbent assay, which targets virus-specific antibodies, immunohistochemistry, fluorescence assay, electron microscopy, Western blot, biosensors, and DNA-based fluorescence nanobarcodes methodology. Each of these methods have their own pros and cons, which can lead to people diagnosing to choose one over the other. For example, some methods can be rapid but require special equipment, but can be insensitive and requires special equipment to be conducted. Now on to Ebola symptoms. What are some of the trademark immunology responses, and how do they make the virus efficient? Some trademark reactions to Ebola virus include viral hemorrhagic fever and a cytokine storm. Cytokine storm refers to various inflammatory cytokines that are produced at a much higher rate than normal. We see a large secretion of pro-inflammatory cytokines and chemokines. These can include IL-1 beta, IL-6, TNF, tumor necrotic factor, CCL3, and CCL4. These are produced by infected monocytes and macrophages. High levels of these are central to the pathogenesis and linked to high mortality rates. In this image, we see filovirus, or the Ebola virus, infecting many cells of the immune system. The effects these can have are an increase of tissue factor, an increase of TNF, some activation, degranulation. Here we see that the filovirus infects the hepatocytes of the liver, which can lead to organ failure and the hemorrhage that we talked about for the viral hemorrhagic fever. We see an inhibition of T cells and B cells and inflammation in the early and late stages. This can lead to an overall poor immuno response, high viremia, which is the presence of the virus in the blood. This ultimately affects many cells and tissues in the late stage of the disease, increases inflammation and increases viral burden to increase the amounts of filovirus.
So what are some treatments for Ebola virus? There are 4 treatment options: large fluid intake to prevent dehydration caused by fevers, blood therapy and immunotherapy to deal with secondary infections caused by weakened immune systems, and finally, experimental drugs. But in 2020, the Ervebo vaccine administered in 2 doses was approved, which can aid in the prevention of infection by certain species of Zara Ebola virus. Of course, a person with the vaccine can still become infected if exposed to such species of Zara Ebola virus, for vaccines are 100% effective. Thank you for your time. We will now begin the Q&A.